everyone, my name is S.cerevisiae and today I'll be discussing the cell wall and aggregation properties of co-evolved isolates of Saccharomyces cerevisiae and Chorella S.cerevisiae. To give you some background, a co-evolutionary system was established by Jennifer Oosthuizen, where yeast and microalgae were provided with mannose and nitrite as the sole carbon and nitrogen source. The yeast metabolized mannose into carbon dioxide for use by the algae, and the algae metabolized nitrite into ammonia for use by the yeast, thus creating an obligate mutualism. Isolates were obtained after 50 and 100 generations and were found to have differences in growth rates, biomass production, and their aggregation phenotypes when compared to the unevolved parental strain. To further investigate the cell wall changes, we conducted phenotypic screens, cell wall susceptibility assays, and microscopy. To very briefly take you through my methods, mono and co-culture screens were conducted using glucose and ammonia as carbon and nitrogen sources. This allowed for the growth of both species and also allowed us to see differences in aggregate formation. Moving on to the susceptibility assays, cell wall targeting compounds calcofluor white and Congo red were used to identify yeast cell wall mutants. Lastly, scanning electron microscopy was conducted to visualize co-aggregate formation between the yeast and microalgae. Moving on to the results, monoculture yeast screens revealed that the parental yeast had the lowest cell numbers when compared to the co-evolved yeast. Co-evolved yeast also formed aggregates in monoculture, while the parental yeast did not. From the algae screens, we see huge differences in growth patterns as the co-evolved yeast exhibit a very long lag phase when compared to the parental. It's also important to note that the algae did not form aggregates in monoculture. As you can see from the co-culture images, there are significant differences in the types and sizes of aggregates formed, and from visual assessment, the algae seem to be incorporated into these aggregates. Cell wall assays revealed that isolate 50.2 and all the generation 100 isolates showed impaired growth when compared to the parental strain. These results show that a cell wall change likely in relation to chitin synthesis has occurred. Microscopy images demonstrated physical contact between yeast and algae cells, implying a mechanism of action where the algae are incorporated into the yeast flocks. In conclusion, it's evident that cell wall changes have occurred over a relatively short evolutionary period. This research also serves as a proof of concept that co-evolution is a strategy to generate strains with a range of phenotypes. Further research needed for this project includes looking at the molecular underpinnings of the cell wall gene regulation, as well as looking at cell wall composition to quantify what changes have occurred to the cell wall. Thank you very much for listening.